For our discussion of the liver, I am going to refer to our PowerPoint just because this is such an important topic and I really want you guys to understand this. The liver has tons of functions. So again, this is one organ system where we could spend an entire semester just talking about the digestive tract and all the accessory organs. But for now, I want to point out that the main function is going to be blood chemistry. There's a buzzword going around right now, especially in the personal fitness world, detoxification. We don't really have to do too much detox. Like a lot of that is just trying to sell people stuff. But our liver is our main detox organ, if you want to think about it that way. It's going to regulate our blood chemistry, and it's kind of our first pass metabolism for a lot of things that come through our digestive tract. Maybe it's the weekends or partying a little bit and ingesting alcohol. All of that is going to interact with the liver before it gets anywhere near the rest of our digestive tract. So this is where processing of drugs as well as healthy stuff like nutrients, that's going to happen at the location of the liver. Another good point is that the liver has two ways of disposing of waste. So this is that term detoxification again, but we have two methods of detox, so to speak. A lot of stuff is going to be excreted in the blood, and this is where eventually things will get filtered down to the level of the kidneys and our renal system. So a lot of waste will exit our body as urine. A lot of waste will also be excreted into bile. And this is where the relationship between the gallbladder and the liver becomes really important. These things are located close to each other because bile is common to both. So bile is going to make its way from the gallbladder and from the liver down to the duodenum via a common passageway, and that is going to be our common bile duct. All right, so I'm going to just push this out of the way for now. There's another image I want to show you that I think will get this point across. So liver circulation is also pretty unique. So you will see this term portal system come up if you take further anatomy courses. And what that means is there is a separate system of drainage for blood to make its way to our liver before it goes on to further parts of the digestive tract. And so that's all this portal system is referring to. So the liver is going to get blood from the portal vein, which, as you can see here, is interacting with all of these other organs of the digestive tract. So stomach, we've got accessory organs like the spleen and the pancreas, as well as our small and large intestine. Those are all getting blood supplied by our aorta, and then their venous output is going to drain to the liver. So this is that first pass metabolism where the liver is going to detox blood before it proceeds to the rest of our venous circulation via the inferior vena cava. We also get blood supply direct from arterial sources. So the liver also has arterial blood coming from our hepatic artery, which we saw when we looked at that liver model as well as the liver lobule model. So two ways we can look at it. Basic stuff to know here is we have the separate portal system taking blood to the liver for additional filtration before it enters into our venous circulation through the inferior vena cava. I have one more slide to show you guys and then we will get rid of that PowerPoint. So let's focus in on histology. The liver is a very spongy tissue and there's going to be a ton of little vessels woven between the actual cells of the liver. And so I want to talk a little bit about the organization. Cellular wise, this is going to look a lot like columnar epithelium. So we've seen that stuff before, lining lumen. In this case, we have a slightly different organization. This diagram does a good job of showing that. Instead of having one central lumen surrounded by cells, we're going to have tons of these little vessels called sinusoids. And these are going to take blood to the actual tissue of the liver so that it can be detoxified, if you will. Though this is a very common pattern that we will see throughout our study of the liver histology. Okay, so we are done with the PowerPoint. Let's just get that out of the way. And now we need our handy dandy human protein atlas. A lot of the histology for this lab is going to be found in the human protein atlas, but we will also refer to the Yale histology site for some other salivary tissue. This is our liver. Let's go ahead and zoom out here so you can see the whole shebang. We are going to be focused today on liver lobules. which are kind of the basic organizational unit of the liver, but we'll also zoom in and talk about what some of these cells mean and what these spaces around the cells mean. So let's go ahead and let's see where the Human Protein Atlas takes us. Just as a disclaimer, our slides might not be the same color. I do believe we have some blue stained liver lobule slides, but this is kind of the basic shape. Every liver lobule is going to have a central vein, and the sinusoids I just mentioned are going to be draining to that central vein. And so that's the basic pattern that we're going to see today. I want to focus today on these liver cell types. We have some different kinds of stuff. We've got neutrophils, which have that immune function within the blood. We again have lots and lots of these sinusoids. So anytime you see kind of a blank space here, or maybe it has lighter stained tissue, that's generally going to be a sinusoid, one of those really little vessels that are helping to both take blood to the cells of the liver as well as take it away. And the main cell that we're going to see in the liver is called the hepatocyte. So most of what we see, these large cells with pink stained cytoplasm and purple nuclei, those are our hepatocytes. In addition, we have these really small guys. These are Kupffer cells, and these guys are playing a large filtration function. What they can actually do is, through the process of phagocytosis, they can engulf foreign materials that we don't want to pass on to venous circulation. Hepatocytes are a little more active, so they are synthesizing different components. They can synthesize bile salts, stuff like that. They can also assist with that filtration. And they do a lot of the actual detox of the liver. So they're going to have that detox function versus just the filtration, which is done by the Kupffer cells. So when you're trying to identify these on a slide, you want to recognize that we are now at the level beyond the liver lobule. We have zoomed in past that point, and now we're looking for just individual cells. There's no central vein to worry about, no liver lobule. Instead, we've got this repeating pattern of hepatocytes. They are larger, they're going to have more prominent nuclei, and then interwoven between them, we're going to have these really small Kupffer cells. And if you see a space like this, that means it is a sinusoid, one of those many vessels passing through to help take blood to and away from the cells of the liver. I want to refer back to our module now to show you what to expect histologically wise. Kupffer cells are hard to find under a microscope, I'll be real with you, so this is more likely to be a PowerPoint question. What you'll need to be able to do is, if I give you the hint that this is liver tissue, I want you to recognize that most of the cells we see here, the large guys with these prominent purple nuclei, these are going to be hepatocytes. The spaces around them are sinusoids. And then typically lining the sinusoid, we're going to have these smaller, darker stained cells. These are going to be our Kupffer cells, helping to filter out blood as it passes through. And then lastly, we have our classic liver lobule. In this case, it uses both names, so sometimes you'll see this structure referred to as a liver lobule, sometimes you'll see hepatic lobule. Either name is acceptable. But I want you to be able to recognize the hexagonal shape we have here. This is the basic unit of our liver tissue, liver lobule. So on this level, we're not dealing with cells. Imagine that this entire lobule is filled with many, many of those hepatocytes and Kupffer cells. And the last feature to know here is we do have a central vein. So all of those sinusoids we just saw previously, they're going to drain towards that central vein. And that wraps up histology for the liver. So make sure you're comfortable with these types of views of the liver lobules as well as this more cellular, zoomed in version. And don't forget about the liver models. So we have two. We've got one showing macroscopic structures, all the big stuff, but we also have a liver plaque showing more of those cellular structures, the types of things that we can see here.